An installed DNA replication experiment. In 1958, two scientists, Matthew Nielsen and Frank Stahl, sought out to expand on Watson and Crick's DNA model and discover how DNA replication occurs in E. coli bacteria. The scientists had three hypotheses: conservative, dispersive, and semi-conservative replication. One hypothesis was the semi-conservative model, depicted on the left. In the semi-conservative model, the two strands separate and each make a copy of themselves. This means after one round of replication, the two new DNA helices consist of one strand of new DNA and one strand of old DNA. The second hypothesis was the conservative model, depicted in the middle. In the conservative model, a new helix of DNA is synthesized so that after one round of replication, one helix is the parent DNA and the other is a completely new strand. The last hypothesis was the dispersive model, depicted on the right. In the dispersive model, DNA material from the parent strands are dispersed randomly between the two daughter helices. They become a mixture of new and old DNA. They began their experiment by growing E. coli bacteria in a nitrogen-15 medium. Nitrogen-15 is a heavy isotope of nitrogen. They did this for many generations of E. coli to ensure that all the nitrogenous bases in the E. coli DNA were now composed of its heavy isotope. They then moved the E. coli into a nitrogen-14 medium, a lighter isotope of nitrogen. After one generation, which was about 20 minutes later, they took some E. coli out of the medium, extracted their DNA, and performed density gradient centrifugation. Density gradient centrifugation is a method where parts of a sample are separated based on their density, where they are centrifuged rapidly or rotated. They repeat this for second and third generation and so on. Here are the results from their experiment. In generation 1, the scientists saw one band of E. coli at an intermediate density. This ruled out conservative replication because they would have seen two bands of bacteria. After the second generation, they saw two bands of density, one at a hybrid level and one at a lighter nitrogen-14 density level. This allowed them to rule out dispersive replication because with dispersive, they still would have seen one band at the intermediate density level. As more generations were tested, they continued to see the nitrogen-14 density level grow, but the intermediate level never went away. Because the intermediate band never went away, they were able to determine that some strands of the nitrogen-15 isotope DNA were still present, which allowed them to conclude that the DNA replication was semi-conservative. Thank you for watching, and I hope you learned a lot.